Hello there, my name is Andy. This past summer, I conducted research in the PageRob lab where I investigated how insecticides can actually alter the evolutionary trajectory of Drosophila fruit flies. And so to start, for a long time, we thought evolution is something that takes place over many, many years. However, that's not true. In some species with very short lifespans, they undergo what is called rapid evolution. For example, in fruit flies, we see that evolution occurs in them in a matter of months, maybe even weeks. And so that makes them a very exciting model system to study evolution in. So to sort of introduce my project, we know that in a given environment, populations face any number of selection pressures. For example, insecticides may be a selection pressure. We know that a population of fruit flies living in the presence of insecticides may evolve insecticide resistance several generations down the line. However, insecticides is just one of many selection pressures. We know that seasonality is actually another selection pressure for fruit flies. For example, natural seasonal fluctuations in the environment can actually determine how dark a fruit fly is, what we call their pigmentation. And so we were interested in seeing the interaction and trade-offs between adaptations to a strong anthropogenic force like insecticides and adaptation to natural seasonal fluctuations. So what really that means is if we have two populations of fruit flies evolving to the natural environment, which we can see through their pigmentation, if we apply pesticides or insecticides to one of the populations, how would their pigmentation change? How would their adaptation to the natural environment change? And so that's exactly what we did. We took 76 fully sequenced inbred lines of fruit flies, some with varying levels of insecticide resistance, and we put them into 12 controlled cages and 10 experimental cages. These cages are outdoor cages, and so the flies will undergo evolution in an environment that's similar to their natural environment. They're allowed to evolve for 6 months, from June to December, and so for the experimental cages, we applied insecticides for several months during this period. Throughout the 6 months period, at several time points, we are taking pictures of the cages and also collecting Drosophila. We take the pictures in order to conduct census estimates to see the population size of the cage. And then we collect Drosophila in order to evaluate resistance level as well as pigmentation. And so to make sure there's no effects of plasticity, we bring the Drosophila back and rear them in a common garden for 2 generations, after which we'll measure their resistance and their population, or their pigmentation, I'm sorry. And so what we expected to see was that for census, we would expect to see a decrease in the number of flies in the experimental cages that receive insecticides because they're dying off. For resistance, we expected to see insecticide resistance increase in the experimental cages. And then lastly, we know that pigmentation changes throughout time for both flies in the experimental and control groups, but we don't really know how insecticides affect the evolution of pigmentation. And so for our first result, we found that insecticides surprisingly did not alter seasonal population dynamics. And so we calculated the population size using using semi-automated image processing of cage photos. And from our estimates, we saw that for both cages, there's not a significant difference. Population dynamics remain fairly similar for both experimental and control cages throughout the entire duration of the experiment. We did not see the population of the experimental cages drop as we expected, which is very, very interesting. We see that the population stayed at the same capacity as the controlled cages. That leads me to my second result. Insecticide resistance evolved rapidly. We can see here from this figure that where the yellow line represents the experimental group and the black line represents the control group, resistance evolved rapidly in the experimental group. So we measure resistance using LD50 to malathion. LD50 is just the percentage, the dose at which 50% of the population survives. Malathion is the insecticide that we're using. And so we see that in the experimental group, during and after when insecticides were applied, resistance or LD50 increased rapidly for the experimental group and even remain in the population after the insecticide period. This may provide a partial explanation to why the population dynamics did not change. Because insecticide resistance evolved so rapidly, we see that the population evolved in insecticide resistance and were able to survive the insecticides during the time where the insecticides were applied and afterwards. And lastly, our most exciting result is that insecticide pressure altered the seasonal evolution of pigmentation. So this is exciting, and I'll tell you why. Because we see that throughout time, pigmentation is changing. So for both control and experimental treatment groups, we see that pigmentation is changing between each time point. However, further that during our August 25th time point and our September 7th time point, we see that pigmentation is significantly different for the experimental treatment group and the control treatment group, which means that somehow insecticides are changing the pigmentation of flies. August 25th and September 7th being the dates during which pesticides were applied or right afterwards. And so this is very interesting. And so I sort of want to bring us to our conclusions now. The first, populations can rapidly adapt to overcome strong selective pressures. We see that with our first two results, how the population dynamics did not alter and how insecticide resistance evolved rapidly. But another interesting conclusion is that we see that adaptations to a strong selective pressure can alter the evolutionary trajectory of another trait. And so we see that the adaptation to insecticides and the evolution of insecticide resistance altered how pigmentation would have evolved. And the reason for this, we think, leads us to our third conclusion, is that populations are constrained to the total number of traits that they can evolve simultaneously, right? So we hypothesize that because the population endured the stress of insecticides, they had to focus on evolving insecticide resistance, and this sort of led to a weaker effect on the evolution of pigmentation, and that's an exciting result. And we're excited to see where the future of this project takes us. Thank you for listening.